Yeah, sorry there was a bit of a delay this week. Had a bit of a cough, so I lost my voice. But I'm on the way back, getting better. So I'm a little bit hoarse today, but I think you should be okay. All right, so I'm going to go over peptide bonds, like protein structure and post-translational modifications today. So everyone should have a worksheet in front of them. So I'll just go through the bullet points first. So that first one, a dehydration reaction, which is a loss of H2O, occurs between the carboxyl group of one amino acid and the amino group of another amino acid. And this results in the formation of a peptide, or you can also call it a covalent amide bond. Okay, so your peptide bond is this thing here. So you have a carbon which is double bonded to an oxygen, and then it's bonded to a nitrogen. Okay, so that's your peptide bond, which is also called a covalent amide bond. Okay, so the second bullet point, the peptide bond has two resonant structures, and this results in the peptide bond having a partial double bond character. To be specific, it's 40% double bond character, but that's not too important. This means that the peptide bond is stronger, so it requires more energy to break. So if you think about it, a single bond would be easy to break, but if you have a double bond, there's two bonds, so you're going to require more energy to break that. And it's also going to be rigid, okay? Because if you had one bond, you could have free rotation, right? But if you have two bonds, then that's going to limit the amount of rotation that can occur if you have a double bond character. Okay, and the peptide bond, it has a trigonal planar configuration. Okay, so that means that all your bonds, there's going to be a bond here, and there's going to be an H bond here and a C bond here, right? So all these bonds are going to be in the same plane, okay? Trigonal planar. And it's going to be mostly in the trans configuration, okay? Which means the R groups are on opposite sides, and this results in less steric hindrance. Okay, so let's draw the dehydration reaction of a peptide bond forming. So your generic amino acid is going to look like this. You're going to start off with you draw the nitrogen first, and it's going to be bonded to, at physiological pH, it's going to be protonated, right? So it's going to be positively charged, and it's going to be connected to your alpha carbon, okay? And then the alpha carbon is going to have an R group attached to it, and it's also going to be bonded to a hydrogen. And then you're going to have your carboxyl carbon, so that's going to have your double bonded O and your OH group, but at physiological pH that's going to be deprotonated, so it's going to be negatively charged, right? And then again, there's going to be the same thing, because you need two amino acids for this to occur. Right, so you've got two generic amino acids, and I'm just going to draw this O in red, and also two of these hydrogens in red as well, so that you can see what's going on. So these two are going to come together. And so these two hydrogens and the oxygen will produce H2O, right? And then you're going to end up with, so you're going to draw the nitrogen first. So you have H, 3 H's bonded to it, and it's positively charged. You're going to have the alpha carbon, so the R group's going to be bonded to the alpha carbon, and then you have your carboxyl carbon here. So now it's going to be bonded to the nitrogen, and then you're going to draw the alpha carbon and the carboxyl carbon again. Okay. So remember that if the R group is on the top, the oxygen, the double bonded O, is going to go opposite that. So it's going to go on the bottom. Okay. So when you're drawing these in 2 dimensions, you want to always draw the O on the opposite side of the R. Okay. So that creates less steric hindrance. Okay. And then the H, there's going to be an H bonded to the nitrogen, and doesn't really matter where that H goes, but usually you just draw it on the opposite side of the double bonded O as well. And then so this one, where would the R group go here? Would it go up or down? Yeah, it would go down, right? Because if it was also on the top, it would clash with this one, because in reality, an R group could be very, very big, right? Because it could be, if it was glutamate, this would be 4 or 5 carbons long, and if this one was also 4 or 5 carbons long, you'll get steric hindrance. So you're going to always try and draw it on the opposite side. So that's the trans configuration that we were talking about. Okay, and it's also going to be bonded to a hydrogen, but a hydrogen's very small, so it doesn't really matter. And then at the end, you're going to have your carboxyl carbon. Okay, so that's your basic dehydration reaction going on with the formation of a peptide bond, and you'll also have H2O forming, which is why it's called dehydration. And you can also call it a condensation reaction as well. Okay, and then so the reason that the peptide bond has double bond character is because it has 2 resonant forms. So your first one here, so you draw the carbon and the nitrogen. Okay, so this is the carboxyl carbon that's involved in the peptide bond, and that's going to be connected to an alpha carbon, right? And this nitrogen is going to be bonded to an alpha carbon, and this is your hydrogen, right? So this is the peptide bond, but it's only like this 60% of the time. So 60% of the time it's like this, and the other 40% of the time it's going to look like this. So there's a double bond between the carbon and the nitrogen, and there's only a single bond between the carbon and the oxygen. And so this oxygen will be negatively charged, and this nitrogen will be positively charged. And you've got the connection, the bond to the alpha carbons on either side. Okay, right. So if you put those two together, the average of those is going to be, so you have your so in reality, most of the time it's going to be a partial double bond character for both of these. So that's what the average is going to look like. Okay. But usually you can just, when you're drawing it, you can just draw it like this, but you should just realize that that's what's going on. Okay, so in reality it's most likely going to look like this. Okay, and if you want to be specific, then this bond here is donating electrons to the oxygen, and then nitrogen has a spare pair of electrons, lone pair of electrons, which is getting donated to that carbon-nitrogen bond. But you don't have to know this detail. It's just if you want to know like the mechanisms of this reaction or this resonance. Okay. And then, so you want to draw the cis conformation of a peptide bond. So in the cis conformation, both of the alpha carbons are on the same side. So here's a carboxyl carbon. It's bonded to a double bonded to an O, and it's bonded to our alpha carbon. Okay, and if it's a cis, it's going to means that the alpha carbon of the nitrogen is also going to be pointing on the same side as well. Okay, and then since the alpha carbon has the R group coming off it, you'll have the R group coming off that alpha carbon, and you can see that if they were both cis, if you had it in a cis configuration, there'd be steric hindrance around these R groups, right? So this is less likely to occur. The only situation where it doesn't matter whether it's in the cis group is if this was a glycine molecule. So remember that glycine, the R group is just a little hydrogen, so then there would not be any steric hindrance. And in the trans configuration, you'd have your alpha carbon on the bottom here, so then your alpha carbon would be on the opposite side for the nitrogen, and then here you've got no steric hindrance, okay, because these R groups are far apart from each other. Okay. All right. So next page. So for proteins and polypeptide chains, the first bullet point, amino acids joined together form a polypeptide chain. Once an amino acid is incorporated into a peptide, it is now called a residue because that's what's remaining of the amino acid. And if you join two amino acids together, that's a dipeptide. So then 3 amino acids joined together should be a tripeptide. And a polypeptide chain can be any length, and the longest ones can be up to thousands of amino acids long. Okay? So we're just quickly going to draw a tripeptide below. So I just like to start off, if I'm going to draw a peptide, I like to draw the backbone first. So the backbone is just the nitrogen, the alpha carbon, the carboxyl carbon, nitrogen, alpha carbon, carboxyl carbon, and nitrogen, alpha carbon, and carboxyl carbon. Okay, so we've drawn the backbone. So this here, this is residue 1. This here is residue number 2. And this here is residue number 3. Okay? So we've got 3 peptides, 3 amino acids there. So it's a tripeptide. And then you can add in your hydrogens and your R groups. Okay, so the one on the end is going to be positively charged. Okay, and then we're going to add in the R group for the alpha carbon and the hydrogen. And then this one here, there's going to be a double bonded O, and remember the O goes on the opposite side of the R, so it's going to be pointing downwards. And then the H for the nitrogen can go either side, but I just like to draw it opposite the O again. And then since the R was up, the R group for this one is going to go down, and the H group will be up here. And then so the double bonded O for the carboxyl carbon is going to be opposite the the R group. So it's going to go up this time, and then the hydrogen can go opposite the O. And then for this last one, the R group will go upwards, and the hydrogen group will go down. And then for the last one, since that's the C terminus, it's going to be negatively charged. Okay. So this side here, this end here is called the N terminus, right? And this side here is called the C terminus. Okay. So you can see that only the terminal amino group and the terminal carboxyl group is what's the word, ionized. Okay. Yep.
For the C terminus, when drawing it, does the orientation of the double bonded O versus the single bonded oxygen matter?
No, that doesn't really matter. Yeah. Yeah. But you got it. Yeah. So you always have to remember that this is just 2D as well. So in reality, these are going to be kind of pointing in and out a little bit. And this, remember that this peptide bond here. So remember, this is the peptide bond. Okay. That's the peptide bond. And so this here, so yeah, and then this one here is also the peptide bond. Remember that the peptide bond is actually trigonal planar, so this isn't actually the correct configuration, but just when we're drawing it, we just draw it in a straight line just because it's easier. Otherwise, your peptide would be going all over the page. Okay. So then the next bullet point, proteins are made up of one or more polypeptide chains. For example, myoglobin is made up of 1 polypeptide chain, insulin is made up of 2 polypeptide chains, and hemoglobin is made up of 4 polypeptide chains. Proteins can have numerous functions. They can be structural, like collagen, enzymatic, so you'll have your digestive enzymes like pepsin, amylase, and lipase. So I think you guys will learn all about that later on in Hubs 192 when you do your GI module. And then it can be a signal molecule like insulin, or it can be used for transport like albumin and hemoglobin, or they can be membrane transporters like sodium, potassium, ATPase, which I'm sure you learned about in cells, or they could be ion channels, so those voltage and ligand-gated sodium channels that you learned about in Hubs, those are all proteins that are located in the membrane. So the primary structure of a protein is simply the sequence of amino acids that it's made of. Okay, so that's it. It's just the order of the amino acids. And by convention, for the numbering of the sequence, we start at the N terminus and we finish at the C terminus. Okay, and that's why you always just, so you can see here that this one here is residue 1 and this one here is residue 2, not the other way around. Okay, so we always start from the N terminus and go towards the C terminus. Okay, and it's the order of the amino acids which contains the information required for folding into the higher levels of structure. Okay, so it's the primary structure or the sequence of amino acids that will determine how it folds up into its secondary structure and its tertiary structure, and then if it's got multiple subunits, it's quaternary structure as well. Okay, kind of like how the order of the letters in a word determine what the word means, and then if you put all those words together, it becomes a sentence, and then it can become a book, and so on. So I like that little diagram at the bottom, just telling you the various levels of structure. Okay. And then secondary structures over the page. So secondary structures are either alpha helices or beta strands. So these are formed from a local stretch of residues, and they are stabilized by hydrogen bonds forming between the amino and carboxyl group of the main chain of the polypeptide, or the backbone. So the backbone is this N, the nitrogen alpha carbon, carboxyl carbon, nitrogen alpha carbon. So this here is the backbone of the protein. Okay, that's the backbone, or what you call the main chain. So the hydrogen bonds are going to be forming between the groups coming off there. So for an alpha helix, most of them have a right-handed spiral. Okay, and there are 3.6 residues in a turn, and there is a distance of 5.4 angstroms per turn, and the vertical distance is 1.4 angstroms per residue. Okay, so if one residue is going up by 1.5 angstroms and there's 3.6 residues in a turn, that's 1.5 times 3.6 is 5.4. Okay, I don't know if you guys noticed that. Probably did, but so the side chains are going to point outwards because there's not enough space inside the helix for the side chains. And usually the side chains on one side of the helix will be polar, meaning they're hydrophilic, while the side chains on the other side will be non-polar. Okay, so if you look at the diagram on the right, if you look at the alpha helix from a bird's eye view, okay, so you're looking down on an alpha helix, one side of the alpha helix will have side chains that are polar, which I've symbolized with the letter P, and the other side will have non-polar side chains. Okay. So what that allows you to do is you could have, remember that if you have a cell, this is a cell membrane. Right, and you have your phospholipid bilayer membrane, right? So phospholipid bilayer, and this area here is non-polar, whereas this side is polar. So the inside, the cytoplasm and the extracellular fluid is polar, right? So if you wanted to allow, let's say a polar molecule like sodium, it's going to have a hard time going through this non-polar membrane. So if you wanted to create a sodium ion channel through this membrane, and then we're going to look at this, we'll look at this ion channel. Okay, so it's going to look like this. So if we're looking at it from bird's eye view, so you could have an alpha barrel like this, which is a sort of domain. Okay, so this is one alpha helix, this is another alpha helix, a third, fourth, fifth, sixth. Okay, so the ones on pointing outwards will be non-polar, right? And that's going to allow it to be anchored in the membrane, the non-polar membrane, right? There's going to be hydrophobic interactions which are holding it in place, okay? Whereas the inside is going to be polar, and that's going to allow the passage of sodium ions through this channel in the middle. Okay, so the inside will be polar, which will allow the sodium channel to come, sodium ion to go through. The fourth bullet point there, so they're normally, alpha helices are normally depicted as ribbons or cylinders in protein structure shorthand. And the alpha helices do not usually contain proline because they won't form the correct angle. So proline is the one that's bonded to itself, and they produce too much of a kink. And the other one is glycine, because glycine is the one where the R group is just a hydrogen atom, and that's going to be too flexible. Okay, so I'll quickly draw an alpha helix just so you can see what's going on. So you have okay, so you have your basic alpha helix spiral here. So normally it goes from the nitrogen end is up the top and the C-terminus end is downwards. Okay, so it goes from top to bottom. So we're going to start off with the N here. So we're going to draw the backbone first. So nitrogen, and I'm going right to left here. So nitrogen, alpha carbon, carboxyl carbon, 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 nitrogen, alpha carbon, and so on. Okay, I won't go on to the end. Okay, so this one here is residue number 1. This here is residue number 2. This here is residue number 3. And this here is residue number 4. That's number 5. And that's number 6. Okay. So you've got 6 residues here. So the distance, the vertical distance from here, so one turn is from here to here. And this distance here is 3.6 residues, or 5.4 angstroms, which is 0.54 nanometers. Okay, and so the vertical distance of amino residue number 1, and then the vertical distance of amino acid, the residue number 2, that's going to be 1.5 angstroms. Okay, and each residue is 1.5 angstroms. Sorry. I'm trying not to make Annika angry by saying angstroms. So you can see that in one turn there's going to be 1, 2, 3 and a bit. I haven't drawn it quite right here, but there should be 3.6 residues. So 1, 2, 3 and a little bit of the 4th residue. So that's 3.6 residues in one turn. Okay, and it's going to be stabilized by hydrogen bonds. Okay, so this carboxyl carbon here will have an oxygen that's pointing downwards, and then it's going to be bonded with, so the carboxyl carbon, so this one's important, carboxyl carbon is hydrogen bonded with the amino, this should be carboxyl oxygen is hydrogen bonded with the amino hydrogen of residue N+4. Okay, so the carboxyl oxygen of residue number 1 will be hydrogen bonded to the amino hydrogen of residue number N+4. So if N is 1, then it would be residue number 5. So it's going to be bonded to the hydrogen of this residue number 5, the nitrogen. So you'd have your hydrogen bond here, right? And then residue number 2, the carboxyl group will also have a oxygen pointing down, and that's going to be bonded with N+4. So 2 plus 4 is 6. So it's going to be bonded with the nitrogen of amino acid number 6. Right. So you have your hydrogen bond there, and that's stabilizing this alpha helix structure. Okay. So they like to ask those numbers or talk about those numbers in the progress test. So it's a good thing if you can just memorize those numbers. But it's not too hard because it's just like, just remember that 1.5 times 3.6 is equal to 5.4. Okay. And yeah, that N+4 is important as well. So make sure you remember that number. So a beta strand is a stretch of residues that interact via hydrogen bonds to form beta sheets. Okay, so when you have 2 or more beta strands interacting, they form a beta sheet, and they have a right-handed twist. Okay, so if this is if my hands are a beta sheet, then they're just kind of twisted a little bit, okay, as they go along. So that's just what that means. And and they're depicted by arrows in protein structure shorthand. So the arrows point from the N terminus to the C terminus. And they can be parallel, which means that both of them go in the same direction, so both of them are going from N terminus to C terminus, or they can be antiparallel, which means they're pointing in opposite directions. And so the side chains are going to stick out above and below the plane of the sheet. Okay, so let's say we have beta strands running down our piece of paper here, and so this here, the piece of paper here is our beta sheet. Okay, so this, if the beta sheet is like this, then you'll have amino acids pointing up, and some amino acids, some side chains pointing up, and some side chains pointing down. Okay, and so one side will all be polar. So let's say that this side is polar. All of the side chains that are pointing upwards are going to be polar, and all of the side chains that are pointing downwards are going to be non-polar. And so that's going to allow them to carry out the function. They can also form beta barrels, like I showed you with the alpha helices. Okay. So if you wanted to draw a parallel and an anti-parallel beta sheet, we'll just do this quickly. So start off with the backbone. So nitrogen, alpha carbon, carbon, nitrogen, alpha carbon, carbon, nitrogen, alpha carbon, carboxyl carbon. Okay, so this one's going from, which way will the arrow be pointing for this one? Left to right? Yep, so it's going to be pointing left to right. Okay. So it's going to be pointing left to right. And so on the bottom, you'll have so now it's going the opposite way. So it's going right to left. Sorry, just doing that again. So nitrogen, nitrogen, alpha carbon, carbon, nitrogen, alpha carbon, carbon. Okay, so you've got your okay, so these are these two are anti-parallel. Okay, so anti-parallel. So we're just going to draw the oxygens and the hydrogens here. So we have the carboxyl carbon here with the oxygen, and this one. So the next one, the hydrogen is going to be pointing up, and then here, the oxygen will be pointing up, and the hydrogen will be pointing down. Okay. And this one here, the oxygen will be pointing down. Okay. So then this one here, you'll have an oxygen pointing up, and that's going to form a hydrogen bond here, and the hydrogen will be pointing downwards here, and then this carboxyl carbon, the oxygen will be pointing downwards, and then this hydrogen will be pointing up, and so you're forming these hydrogen bonds, right? So this is one beta strand, and this is another beta strand, which are in an antiparallel configuration, and they're forming hydrogen bonds between them, which are stabilizing that secondary structure. And then we can also draw another one up here. Okay, so this one's also going to be pointing left to right. Okay, a little bit wonky, but so here this one's so antiparallel. So for parallel ones, the hydrogen bonds kind of form at an angle. Okay. So we've got the oxygen going up. Oh, sorry, this one should be going down. So that's going to form a hydrogen bond here. The oxygen will be going up, and that will be forming a hydrogen bond here with the one below. Okay. So it's good practice to try and draw these, which is just try and get it into your head how they go. Okay, and so yeah, you'd have if you were going to draw the R groups here as well. The R group here, one of them would be above the plane of the beta sheet, and then the next one, the R group would be below the plane of this beta sheet. And then the next one, the R group would be above the plane, and same here. So this R group here might be pointing upwards, while the next one here will be pointing below the plane of the sheet. Okay, and they'll always be alternating. Okay, and usually, so if this one was polar, this one would be non-polar, and then this one would be polar. Okay, so that'd be alternating polar and non-polar. All right. So then we'll move on to supersecondary structures. So it's never really clear what the difference is. To me, to me anyway, I never really got the difference between a supersecondary structure, a domain, and a motif. Okay, so to me they're kind of all the same thing, but some people think differently. So if you put a whole heap of alpha helices or beta sheets together, they can form what you call supersecondary structures, and they're going to be connected by loops and turns. So the difference between a loop and a turn is usually only about 3 or 4 amino acids long, while a loop is a bit longer than that, so they could be up to 10 to 15 amino acids long. Okay. Supersecondary structures can merge to form a domain or motif. So the way I understand it is that supersecondary structures are combinations of secondary structures, and then domains are combinations of supersecondary structures. But I might be a little bit wrong there, I'm not too sure. So usually domains are going to have a specific function, like binding a substrate, or they might catalyze a reaction. Some proteins only have one domain, while some can have multiple domains. And similar domains can be found in different proteins. And you've just got a little bit of a diagram there showing the hierarchy of these protein structures. But just take that with a grain of salt, because there's different classifications out there depending on where you look. In some textbooks they'll say differently, but they're not going to be like super anal about it. Okay, and then tertiary structure. So there's three-dimensional conformation, which is another word for the shape of the polypeptide chain. So residues that were far apart in the primary structure, they can end up being close together due to folding. And they're stabilized by non-covalent bonds. So examples of those are ionic bonds, hydrogen bonds, hydrophobic or non-polar interactions, or metal-ion coordination. So if I hold on to a metal ion and another person holds on to a metal ion, then we're both connected due to that common bond. Okay, and it can also be stabilized by disulfide bonds, which are a type of covalent bond. So remember that disulfide bonds are the bonds that form between two cysteine residues. Okay, and then quaternary structure. So the conformation of the protein when all of the subunits are together. So one subunit, so when they say subunit, it means each polypeptide chain, okay? And so a protein can only have quaternary structure if it's made up of multiple subunits or multiple polypeptide chains. And the quaternary structure is stabilized by non-covalent interactions. So if I were to give you an example, myoglobin is a monomer, so it's only got one subunit, so it has no quaternary structure, right? Whereas hemoglobin is made up of 4 subunits, so it's got quaternary structure. Okay, and then they talked about the Anfinsen experiment, which is where they broke all the disulfide bonds that stabilized the tertiary structure, so which caused the protein to become denatured. Okay, so that's an important word, denaturation, or if a protein becomes denatured, it's losing its shape. Okay, and if it loses its shape, then it can't perform its function. And when urea was removed, the protein was able to refold. So that showed that the information required was in the primary structure, but not all of the proteins will be able to refold into its original structure once it's been denatured. Some are going to require specific environments, like they might need the correct pH, or they might need chaperone proteins. So those are proteins that help the folding of a protein, of other proteins. And the sequence of folding has to be in the correct order. I'm not sure if you guys have done origami, but you guys, if you have done origami, you'll know that you have to fold the piece of paper in the correct order. Otherwise, it's not going to end up in the right shape. And it's if you have a simple protein, you might be able to just crunch it up and get the right shape. But if you have a very complicated protein, everything has to be in the correct sequence. So protein folding, hydrophobic residues are going to move away from the aqueous environment because they don't like the water. And that's going to initiate the folding. And then the secondary and supersecondary structures are going to form. And then the supersecondary structures come together to form domains. And then the domains move close to each other. And then the non-covalent interactions stabilize the new conformation, as well as covalent disulfide bonds. So in the past exams, in the finals, they like to ask questions about primary, secondary, and tertiary structure, and they like to use myoglobin or hemoglobin as an example there. So just make sure that you understand what primary structures, what secondary structures, and how to describe all of it, and what tertiary structure and what quaternary structure is as well, and how the order, what initiates that protein folding. Okay. So protein misfolding, the normal prion protein, so that's PRION. The prion protein is a protein that's normally produced in your brain, in the central nervous system, and it's required for normal brain function. But if you get exposed to abnormally folded prion protein, it can cause the normal prion protein to turn into the bad form. So normal prion protein is mostly alpha helices, but the bad prion protein has more beta sheets. So I remember this by thinking B for bad, and they do ask about it in the progress test. So these misfolded prion proteins, they're more likely to aggregate and clump up, and if you have big clumps of protein in your brain, that's going to result in dysfunction, and you can end up dying from that disease. And the scary thing is that there's no cure for it. Okay. So quickly, I'll go over post-translational modifications. So a PTM for short is any modification that's made to the protein after it has been translated. So translation is the production of the peptide by the ribosome using the mRNA that was transcribed from the cell's nucleus. And most proteins in our body require post-translational modifications in order for them to function properly. So if you've got a protein that's just been translated and it hasn't been modified, it's not going to be able to do its job. So a few of the PTMs that you should know about, I've listed there. So phosphorylation, so that's the addition of a phosphate group to the hydroxyl group of a side chain on the protein. Okay, so if you have let's say we had a serine molecule, so we had a hydroxyl group on our protein, on our amino acid. We can replace the hydrogen with a phosphate group. Okay, so these oxygens are negatively charged. The phosphate group is negatively charged. And if you're introducing negative charge, that's going to change all of the dynamics, the electrical dynamics between the proteins. So if you have, let's say, the next let's say this one here, it was, let's just say it was a positively charged molecule. So then you've introduced a negative charge here, so that's going to want to get closer to the positive charge, and that's going to change the shape of the protein, right? So by introducing these negative charges, or by phosphorylating the protein, you can induce a shape change, a conformational change, and that's going to change the activity of that protein. So it can either turn it on or turn it off. And the proper name, the proper word for activation, or for turning it on, is activation, and for turning it off, it's called inhibiting a protein. Okay, so that second bullet point there, enzymes that add phosphate groups are called kinases. Okay, so Kinases. So kinase means that you're adding a phosphate. So any word that ends in A's means it's an enzyme. Okay. Okay. or it can be called a phosphorylase. So the difference between a kinase and a phosphorylase is a kinase is using ATP to transfer is taking the phosphate group off an ATP molecule, while a phosphorylase is just using a phosphate from anywhere. Okay, so, and then you've got carboxylation, hydroxylation, and peptide bond hydrolysis. Yeah, so they used to be important, but I got told that they're not really going to examine them that much this year. Okay, so that's it for today. I'll be back tomorrow night at 8 o'clock, so see you guys then. Thanks. All good.